the importance of our new paper that appeared in ASN Neuro last week shows for the first time that amino acid fermentation can also participate in the dysregulated growth of the tumor cell. And the amino acid that we're speaking predominantly about is glutamine. And we tested all 20 amino acids. We interrogated the cancer cells, both mouse and human glioblastoma cells. We interrogated them to ask what fuels could they use to maintain their growth. So the way we do this is very simple. We just take the tumor cells and we grow them in saline, salt solution. And of course, there's no food. There's nothing for them to eat. And then you take and time them to see how fast they die with no food. And mouse cells, because they have a very high basal metabolic rate, 7 times faster than that of the human, they die quick when you take away their fuels. Mouse cells will die in 24 hours. Human cells will die in about 72 hours, 48 to 72 hours with no fuel, just in what we call a buffered saline solution. And then what we do is we simply add individual amino acids back, and we see whether or not the viability of the cells is improved. And what we see, we do this both in the presence and absence of glucose. So glucose alone will keep the cells alive longer, but then they die because they don't have any nitrogen source. And we do it without glucose. So we just do it in pure nothing, and then we add glucose, and then we add individual amino acids back. And what we see is when we add the amino acid glutamine, these cells explode in growth capacity relative to all other amino acids. Now, we found a little bit of stimulation from glutamate because it's the first product that glutamine is metabolized to. But it's not nearly as powerful as glutamine. None of the other amino acids had anything come close to how powerful glutamine is. So the question is, and when you have glucose and glutamine, the two fuels are synergistic in providing all of the metabolites and all of the energy needed for rapid dysregulated growth. So the question is, how is glutamine able to facilitate the growth of the tumor? Is it respired through the respiration? It's called an anaplerotic respiratory process, where the carbons of glutamine enter into the TCA cycle to produce reducing equivalents, allowing oxidative phosphorylation to operate, producing energy. Or is the glutamine fermented, which is generating energy through mitochondrial substrate level phosphorylation in the succinyl-CoA ligase step within the Krebs cycle or the TCA cycle? So what we did, and why this paper is so important, is we were able to test this hypothesis many different ways. So we would take all of the glucose away from the tumor in the presence of oxygen and then give glutamine, and we found that the cells could survive with oxygen and glutamine in the absence of glucose. But then that doesn't tell us whether the glutamine is fermented or is respired. So we repeated the experiment, this time removing all the oxygen. We grew them in a deep hypoxia in the absence of glucose. And we still got ATP production in these cells. Yes, it was significantly reduced, but it was still being produced. ATP was still being produced in the cancer cells in the absence of oxygen and in the absence of glucose. So the question is, where is that ATP coming from? So we think it's coming from the substrate level phosphorylation in the TCA cycle. And we were able to show that when we use labeled glutamine, C13 glutamine, we were able to see C13 succinic acid as a waste product. That's the end product of the glutaminolysis pathway. Clearly, the glutamine is coming into the cell, metabolized to glutamate, to alpha-ketoglutarate, to succinyl-CoA, and we found labeled C13 succinate, the end product of the glutaminolysis pathway, meaning that the fuel is being dumped out. It's not anaplerotic to any extent. It's being dumped out as a fermentation end product, showing basically that these cells are using glutamine in a non-oxidative way. Another experiment was we used cyanide, which blocks oxidative phosphorylation, and we were still able to see significant ATP in the absence of glucose in the presence of cyanide, using glutamine as a fuel, and we were still able to see this succinic acid produced, clearly showing we disconnected oxygen and oxidative phosphorylation from a second major pathway in the mitochondria producing energy. This is unbelievable. So this is the missing part of Warburg's central theory. Warburg had everything right, most everything, not everything. He was correct on knowing cancer was a mitochondrial metabolic disease, but he did not know there was a second major fuel that these tumor cells could ferment together with glucose. So this now opens up the whole field of cancer to know these tumor cells are dependent on two major fermentable fuels. So we're able to clear up the misunderstanding of Warburg. We're also able to say that the oxygen consumed in the tumor cells is not used predominantly for ATP synthesis.